doing single cell years. So yeah, first thing I'm going to be talking about is, well, I have an example. So you've done your single cell RNA-seq. You've got some clusters. You want to identify marker genes defining a cluster. This is like a classic use case. So for example, in Seurat, we want to identify the marker genes for this cluster here that's in red. What Seurat's doing under the hood in their find markers method is they're actually just running a Wilcoxon rank sum test on the difference between gene expression of each gene in red versus the blue. And so the output is this volcano plot here with gene expression ratio or log fold change on the x-axis and your FDR adjusted p-value logged on the y-axis. And here we go. We've got BUB1 being a checkpoint kinase gene, being the strongest marker for this cluster. Makes sense. They're actively cycling cells. Great. Wow, that is a really high p-value. It's like 10 to the negative 40, right? And that's after FDR adjustment. That's crazy. Do we trust this? And that's where we get into this double dipping thing. So double dipping is a term coined by a lab that I'm going to be talking about extensively, but the real problem that this encapsulates is this concept of selective inference. So these slides are blatantly ripped off from Lucy Gao and Daniela Witten. Lucy Gao is now a prof at University of British Columbia, I think, and Daniela Witten is a prof at University of Washington, and they've been sort of the strongest advocates for not making this mistake anymore. So yeah, in this slide, they've got a random sampling of 100 observations with 2 features. So we've got 2-dimensional data here. They've clustered it, and if you do a test of difference of means between the clusters, you get really strong p-values. Shocking. But that's essentially what we're doing when we're looking for these fine marker genes and doing a differential expression test, right? So a common, alternative might be, let's split our data and we won't do the inference on the set that we clustered on. So we've got a training set and a test set. We learn from the training set, we classify the test set so that we didn't do our clustering on the same set that we're doing the inference on, and still we have a p-value that encourages us to discard the null hypothesis, which is not true. So what's the problem here? The problem is that we're testing differential expression between clusters using the same information that we use to define the clusters. This is similar to overfitting linear regressions, which is by selecting only relevant features, and that's actually where we're going to borrow the solution from. So this concept came up in the linear regression overfitting literature. Jonathan Taylor's group, I believe, was into that, and they talked about this selective inference issue, and they said, okay, you can fix this problem if you condition your null hypothesis on the aspect of the data that defines the groups. So in this example here, you're not asking, is the difference in means between these 2 clusters more than we'd expect by chance, but we're asking, is the difference between these 2 means between these 2 clusters more than expected by chance, given that we've got 3 clusters in this data, sort of thing. So you condition it on what you've done to the data, and if you do that, you go from having non-uniform null p-value distributions to uniform null p-value distributions, which is what we should have when the null hypothesis is true. That conditioning step turns out to be hard, and that's been a source of a lot of research. So the Witten lab has worked on that for both hierarchical data and k-means clustering. The drawbacks to both of these for our applications is the way you clustered the data matters. So in order to define the condition, they need it to have been either hierarchically clustered or clustered with k-means, neither of which we use in single cell RNA-seq anymore. Hierarchical clustering is too slow, and k-means is it's just not as good as the graph-based clustering that is commonly used, like the Seurat method, for example. Also, these methods are not designed for feature-specific testing, the individual genes case, which is the case that we use in single cell. So despite the Witten Lab being the best advocates of this problem, they don't actually have a solution for us, unfortunately. But there is a solution out there. This paper, which made absolutely zero splash when it came out in 2019, unfortunately, seems to have the solution. So this is a test designed for testing differential expression of genes between clusters, and they note that clustering tends to generate linearly separable features. So if you learn the line that separates two clusters for each feature, and then apply that to all features, you end up with this hyperplane, which is a fancy way of saying a line that separates things in multidimensional space. But yeah, now you've got that hyperplane, you can define, instead of using the normal distribution or a t-distribution or whatever for your hypothesis test, you can define a pair of truncated normal distributions that are split by that hyperplane, and that is your conditioned null. And so if you do that, you regain a uniform p-distribution when the null is valid, at least according to their paper, which, as I said, has made zero impact. So we've got no, third-party peer review on it, unfortunately. And as I said, I am not a statistician, so I am just running on faith here. But now you know. So yeah, the logic is you split the data. You do your clustering scheme, and it can be whatever you want because the clustering scheme doesn't matter, because then you do a hyperplane estimation, and then you label the new data using that hyperplane and do your TN test. I know the data splitting thing sounds like something we said didn't work, but it's this conditioning on that hyperplane that makes it work. Okay, so back to the example case. Do we trust these p-values? No. But does it matter? And this is in my mind the reason why I haven't gone back and run the TN test on all my data, is that personally I'm not announcing, oh yeah, BUB1b has a p-value of this and therefore it is the best marker gene. If you don't care about that p-value because, say, you're just trying to annotate the clusters using GSEA, which you just use a ranked list of your most significant, or well, all of your gene expression in that cluster versus others, if you're not reporting these p-values, then it doesn't actually matter. That being said, if you are, you definitely need to go and solve this problem. So now you know. All right, so the next one is easier and harder at the same time. So it's this concept of pseudo-replication bias, and it has been noted that sometimes when we're doing case control studies in single cell, we use each cell as a replicate, which is not a good idea because the cells are non-independent. The subjects that we took those cells from are, in fact, the independent things. Yes, Shimini. There's a question in the chat. I will find the chat. Oh, there's actually lots of talking on the chat. Okay. Do you know how fast this method is? I assume that's TN test, and no, unfortunately, I haven't tested it. I don't. It's probably, well, I can guarantee you it is orders of magnitude slower than doing the fastest implementations of the Wilcoxon rank sum test. For example, that Presto implementation of the Wilcoxon rank sum is insanely quick. So this is going to be significantly slower, which is why I would argue that, unless it matters to you, maybe don't worry about it, but now you know, so that if it matters, you can take the time and actually do it.
Thanks, Brandon. Can I ask a follow-up question?
Yeah, so this group noted that, hey, we got to stop treating each cell as a replicate when we're doing case control studies, or any study where we're comparing organisms, because that's not the case. So being statisticians, they didn't have any pretty pictures, but they did have a solution, and their solution is conceptually quite easy. You just consider the sample replicate. So they argued that the best way to do that is to introduce a random effect term relating to what sample each cell came from in your general linear model. So that's assuming you're using something like MAST, which is this two-part hurdle model they're talking about. They were pretty fascinated by it. So they tested MAST, this two-part hurdle model, with and without this random effect term, and then reported the results in this beautiful table because they hate their readers, and noted that the type 1 error rate, which is your p-value essentially, your type 1 error rate should be what your p-value is, was 0.5 or 0.6 or wildly high when the p-value was supposed to be set at 0.05. And in fact, it goes up, as is not obvious in the table, but I took the table's numbers and made a plot here as the number of cells goes up. Because we're using each cell as n, the estimated standard error is going down, so our significance seems to be going up. They added this random effect term, and suddenly type 1 error rate is controlled. I highlighted anything where type 1 error rate was within 0.02 of the target p-value of 0.05. And so yeah, type 1 error rate is controlled. They also tested pseudobulk, and pseudobulk is exactly what you think. So for each sample, you just take a mean, or you sum the gene expression values of all the cells in that sample, and then you do whatever test you want between those. So you go from having, say, in this case, 500 cells to having 5 testing things because your n is 5, and you do your pseudobulk. And that sounds like you're throwing away a lot of information, and their argument was that, yeah, you are, and it ends up being overly conservative as a result. But yeah, it is an easy alternative to the thing they're proposing. As a quick aside, they can hear you, but someone says something out loud. Yeah. Oh, okay. I have no idea how to do that.
Did you increase your?
Oh, I can increase my volume, but I'm not sure if that makes a difference because it's plugged in over there. Does someone want to talk and we can figure out the solution to this?
Hey, Brandon. Can you hear me?
Hey. Okay, there we go. That works. Hi, Delaram.
Hi. I just wanted to clarify something. So for the double dipping problem, or I guess it's also true for the pseudo replication bias issue. Are we certain that all features or genes are equally affected by the p-value inflation? So can we be confident about the order when we're dealing with these issues?
That is a great question that I actually don't know the answer to. And I realized that as I was practicing this talk and went, oh dear, I should have tested the TN test to find out. So that's a good point. If you go and do the TN test, or, if you go and do your TN test, you should compare it and let us know on the Slack group. But yeah, that is a really good point. It would be nice if the rank order of significance was the same. And yeah, I don't know. I'm trying to think of whether, so all the differential expression testing benchmark studies, I'm not sure that any of them actually looked at between cluster things. So none of them, picked up this double dipping problem. Because they would have highlighted that the rank order changed, but I'm not sure that anyone did. So yeah, the answer is I don't know.
Okay, cool. Yeah, it would be cool to evaluate it. Thanks.
Yeah, maybe I'll do that before I leave. All right, so as an aside, it turns out that a subset of the single cell community has been not making the pseudo-replication error and using these random effects for a while. Those are probably people that, are statisticians. I'm highlighting this blog because I think if you're, maybe not so much anymore now that single cell community has matured, but the best solution in the early days was to find people who you liked their science and follow their personal blogs. This guy's personal blog snuck into a random blog post about something completely different. He actually has the random effect term built into his linear model here, and this was before the Langefeld paper. Anyway, the Langefeld paper and the random effect term being better than pseudobulk raised some stink, at least with this group, who argued that no, pseudobulk is in fact better, your metric was wrong. And then these guys replied, and there was some drama. So yeah, they're like, the early days of single cell was fun for drama. And this wasn't even early days, this was last year. So single cell is still fun for drama. You just gotta like keep paying attention to the margins where people are arguing about what method is best. And if you follow Lior Pacteur, he argues that everything's wrong and we're all idiots. So he's always great for good drama. But anyway, I'm not gonna go into that because frankly, from a non-statistician's perspective, like who cares? They're both pretty darn close, whatever works better. I love this paper, which actually came out around the same time. And this group, which is actually a bunch of, single cell types, as opposed to statistician types, empirically discovered this same problem. They were doing a benchmarking test of differential expression testing. And they were looking at, they figured the best way to benchmark it was to get papers where people had done single cell and bulk on the same, asking the same question. So they gathered up as many data sets like that as they could find, and then looked at concordance of differential expression results between single cell and bulk using a bunch of different tests. And all the winning tests were these tests meant for bulk, but they sort of added an application for single cell, like EdgeR and DESeq and Lemma. And they noticed that they'd all built in a pseudo-bulking step. So they took your sample things and just pseudo-bulked the samples, and they performed best. So they went in and manually removed that step, and those same tests are now here in gray. So they were in red, they're here at the top. In gray down here, suddenly they don't perform well at all. So the pseudo-bulking was the thing that mattered. Furthermore, they noted that testing methods without pseudobulking seem to have this strong correlation between gene expression levels and whether they were going to show up at the top of your differential expression testing results. And it turns out that if you take away the pseudobulking step, that shows up in EdgeR and Lemma and DESeq as well. So yeah, the pseudobulking thing really matters. The other neat thing they noticed is that you can't get away with faking replicates. So they tried this concept of just, okay, so we don't know which cell came from which replicate, which I'm going to talk about more because that is the problem with this very obvious solution. But maybe we can just randomly make replicates. We know how many animals we sacked to run this experiment. So let's just randomly assign cells to that number of animals. And there we have the solution, but no, the variance is actually less with the pseudo replicates than with actual biological replicates, as you can see here. And that was the case in nearly all of the data sets they looked at. So that's a problem for doing that. We'll talk about that more in a sec. But yeah, so that gets me to my concern with this whole thing, which is how do we apply this knowledge? Because I know, at least in the single cell data that I've worked on, sometimes, if I'm lucky, I know how many organisms were contributed to this data, but I do not know which cell came from which organism, because in your droplet single cell, you put everything into one lane or 2 lanes, and you run it all because it's expensive. So if all your replicates are in one run, great, you've got no batch effect, but you also have no ability to distinguish between replicates. And of course, that only works if your case and control are genetically distinct, which, maybe they are because you labeled them, or maybe they're not, in which case you didn't do this. Or you could do all your replicates in individual runs, which no one does because that is wildly expensive. And not only is your casing control confounded with batch, but replicate is now confounded with batch, so that's not great either. Finally, you do your case and control individual runs, which unfortunately happens more often than it should because treatment is now confounded with batch, which makes things hard. But sometimes that's the only way to know which one was treatment and which one was control if you don't have some genetic marker for which is which. But you still have no ability to distinguish between replicates. So the statisticians were like, hey, this is an easy to fix problem, but practically speaking, the way we design our experiments, this is not. So there are some solutions. In one of them is in advance, this concept of cell hashing, which is not a new concept, but it's not that popular because it's hard and it's probably expensive, I don't know. It's this idea that you use the site-seek method of barcoded antibodies to label cells in some way, and you just label cells with which sample they came from so that you can pool them all together and still get barcoded, this cell came from this sample in your analysis. So that's great. It eliminates your batch effect problem. You can track samples. It's absolutely fantastic, but not everyone does it because it's hard. Like it adds a lot of complication to your library prep situation. So if you have that option, awesome, but not everyone does. There is a post hoc solution that I stumbled upon when I was looking at this benchmarking paper for batch effect integration differential expression. These guys looked at batch effect correction methods. They looked at, doing a differential expression with a covariate for batch. And then they said, or we could just do meta-analysis or just be naive and just throw it all in and see what happens. And it turns out that they tested it on a variety of different sort of experimental contexts in terms of like full length or high throughput single cell, but the high sparsity, low depth context that they were testing, which is the equivalent to your drop-seq type thing, the things that performed best were actually, and I've highlighted them here, although you probably can't see them, but you can go to this paper and read it, were either meta-analysis or just naive Wilcoxon rank sum test on the raw, like unbatch corrected data. So there you go. I think the key to that is having enough samples. So not just 2 batches, but like 20 batches. So your best bet is if you've done some case control studies, but someone else from another lab has also done some case control studies, bring in their data and you can do sort of a meta-analysis, and then the batch is getting sort of coming out in the wash because you've got enough different batches. However, there is also this one other paper, which is complicated, but they argue that the obvious problem here that we're using non-independent replicates, the cells, rather than independent replicates, the subjects, isn't necessarily the problem, and that the problem could be distributional misspecification, which is tricky, but the logic is that within your cluster or whatever you're doing your differential expression test on, it's not necessarily perfectly Poisson distributed or whatever your general linear model assumes. And that can have effects on your estimates of variance. So distributional misspecification doesn't affect point estimates like your magnitude of difference, your log fold change, but it does affect error. It causes your variance to be, your estimates of variance to be less than what is truly out there. But it turns out that increase in variance, because you screwed up which distribution you're going to test against, has a maximum theoretical amount, which is the sandwich variance, and that can be predicted by these robust standard error estimators, also known as sandwich estimators. And there's actually an R implementation for sandwich estimators, because this is a thing that's been around for like 40 or 50 years now, which is great. So their argument is that you can do your Poisson or negative binomial general linear model test, so like not masked, but same sort of concept, but just use the sandwich estimator, and that should control your type 1 error rate. And so they show that here. They've done like a Poisson test, and then they've done it with their robust estimator, and the p-value distribution gets closer to the null or the uniform distribution in the case of a null test. It's still off a bit, but it's better than without the robust tester. Negative binomial, it's considerably better when it's robust. And then they compared it to pseudobulk methods, and they say the robust is even better than the pseudobulk methods, although that does depend on the number of samples you have. So this, I highlighted the one with 8 replicates, which I thought was a little more close to our general realities. They also tested on some data sets with hundreds of replicates, and the pseudobulk methods perform as well as their robust error estimator or sandwich estimator implementations on those. So yeah, the nice thing about this is you don't actually need to know where your samples came from for this, because you're not correcting for pseudo-replication. So maybe this is an alternative solution. Anyway, I've kind of blazed through this because nobody is yelling at me about things. So that gives me time to talk about my own little pet project, which matters considerably less than anything we've talked about so far. But I don't know if anyone can see this or whether I need to zoom in. A couple years after I brought it up as an aside in a random paper that I published, Leo Pactor's group did note it in this teensy little bioarchive preprint, which they made into a fun joke about it in the title, where they have log x plus 1 and log 1 plus x formulae contributed equally to the title, which I thought was cute. Anyway, the thing I noted is that, so when we do our differential expression, or when we do our normalization, actually, we log transform our count data for reasons. It's a variance stabilizing transform that's just, it's happened historically in bulk RNA-seq, and so we keep doing it. There's a good reason we do it, but I'm not going to get into that. But when you log transform your data, and your data is 0, that doesn't work. So the solution has always been to just add 1, because in bulk RNA-seq, the counts were in the thousands, so adding 1 was inconsequential. But in droplet RNA-seq, adding 1 is consequential because the counts are in the tens or even the like ones. And so it's not a problem if you're just looking at abundance of gene expression, but once you do comparisons, the ratios get squashed. And that, in this figure that I didn't fix because I've been roasted for it before and should have fixed, but if you're doing comparisons of small versus small, so this would be like 1 versus 0 counts or 2 versus 1 count, you'd expect your ratios to fall along this line, but they end up going way off it real quick, because if you imagine 2 versus 1 is a full change of 2, but if you add 1 to both those things, now you've got 3 versus 2, which is a full change of 1.5. So that's a big difference right off the bat. Okay, so the solution is easy. You add a ridiculously small pseudo count, so 1e to the negative 99, but now you have this other goofy problem, which is that if you do have a divide by 0, which is quite common because we've got lots of zeros in our data, then your fold change becomes goofily large, like in this case, a fold change of like 300. And this is sort of a bit of an up to you thing because no one can tell you the answer here, but my thinking was 0 in droplet data isn't necessarily 0. It's just that in that cell, we didn't count that gene. It might not be expressed at all, but it might be expressed just slightly less than the window of sensitivity in which we can actually count. So it's like 1 less than 1 count, which on a normal scale would be fine. That's 0, but on a log scale, it's not fine. And so in order to capture that, I thought maybe we do a pseudo count of 1 over the number of cells, because that is one less than the smallest number we could possibly get in our data, which ends up bringing your fold change when you're dividing by 0 to a more reasonable, say, -10 to -20, in the same sort of range as the rest of your fold changes. So that was my little fix for this problem. This thing is completely inconsequential except for magnitude reporting for differential expression, which some people get bent out of shape about how small the magnitudes of differential expression are in single cell. And that's for two reasons. One, we're not sequencing deep enough to get many. Essentially, our counts are, is it there or not, really? So if we sequence deeper, we'd get higher magnitude differential expression, but that's expensive. And two, this problem, so we're already making counts that are small because we're not sequencing as deep as in bulk, smaller because of this silly little math error. Anyway, that's all. I should check to see if there's anything in the chat. There isn't. So I'd just like to thank everyone. The Bader Lab, specifically this handful of folks from my time in the lab, some of whom aren't actually here anymore, but have all worked on single cell and done cool stuff. The Panoramics team for making this happen. The previous iteration of this from way back in the day where we all just got together and groaned about how no one knew what they were doing, and the Princess Margaret Genomics Center for hosting that group therapy session way back in the day. Thank you, everybody. And let's, oh yeah, I do have one last slide that I'm just gonna leave up if there are any questions. There's a couple of benchmarks from differential expression testing. One was the original benchmarking paper by Sonesson and Robinson, and follow Charlotte Sonesson. She's been doing benchmarking on a bunch of other stuff. She's got all sorts of good benchmarking papers. And then this one was kind of interesting because they did an evaluation of the whole analysis pipeline with the metric being differential expression testing and noted that normalization matters the most. So I hope you guys were here last week for that talk because that's the most important part. And then here are a couple of just general single cell guidebooks, an older one and a newer one, both of which are quite good. Thank you.
Do we have any questions from the audience? You can unmute yourself or you can type in the questions.
The sandwich paper is arguing that pseudoreplication isn't relevant. They are specifically saying that, yeah, conceptually it absolutely should be. It's wild, but I think their argument is not, oh, it doesn't matter. It's that this, by addressing the distributional misspecification, you end up solving the problem a different way. The logic there is twofold. One, they argue that autocorrelation between cells isn't as big a deal as we think it is. Maybe they're not as well correlated as we think they are, which, based on the fact that we can cluster them, I'm not so sure about. But it actually, I think, might go back to this finding, which is that either way, what we're really getting down to is that we're not properly capturing the variance. So we don't have a proper estimate of the error because essentially, right, any frequentist type expression test is you're just how far outside of the like expected error is your value. And if it's far enough outside, then we don't think it happened because of error. And so it must be real, essentially. And so whether we've defined that error by having the appropriate number of n, or we've just used some other math to come up with the right error, and that's what the sandwich estimator is doing, is coming up with a bigger error than we'd expect based on the number of n, because it's arguing that the error based on n is the distribution, and we got the distribution wrong, so the error is going to be somewhere larger. So I think it's just they're coming at the same problem from a different perspective, which is good for us because, yeah, in my data, I don't know where the samples came from. Does that help, Delaro?
How do they know that autocorrelation is not a big issue?
Yeah. I think they say, again, I think they say it is a big issue.
Like what I mean is that I guess it exists and you can't deny that. Like I guess I just don't fully understand, and I guess my knowledge of statistics might be not
Yeah,
enough.
I mean, neither do I, but I think like watching or like reading over these papers again recently, like when I first read that paper, I was like, I have no idea, and this doesn't make any sense, because obviously it's pseudo-replication is the problem. Like that's a super clear problem. But then like these guys like noting that it's not just the N, because like I was thinking, oh, great, the solution is what these guys tried. You just make up replicates so that you tell the distribution, yeah, it's going to have this many replicates, problem solved. But that doesn't capture all the error, and you actually need to capture all the errors. So you either need the true replicates or these guys thing, which is just getting to the real. I think the argument is the fundamental problem is the error estimate is smaller than it should be. And it's smaller than it should be because of pseudo-replication, absolutely. But if you can come at it from a different direction that also fixes that error estimate, maybe you're halfway there. Maybe you're not. Maybe they're full of shit, and these values, I mean, the reason why the Poisson robust doesn't get you to a null p-value distribution the way you'd expect it to if this were completely solving the problem, maybe that's because it's not. These guys are wrong, and it's just that you're using a sandwich estimator to increase the error so it's getting better, but like in a randomly good amount. So sometimes it works and sometimes it doesn't. I don't know. I'm sort of hoping that I keep like searching this paper on Twitter and stuff, waiting for someone to be like, no, this is dumb. But no one's done that yet. So I'm stuck.
Yeah, from practical perspective, I understand if they argue that, you know, in practice, it's not as big of an issue, but it seemed to me that they're kind of addressing different problems. But I haven't read the sandwich paper, so I'm not sure. But another question that I had was that, do you know, like, could you go back to the variance scatter plot that you were showing?
This one.
Yeah. So do you know what technology is the data coming from? Is it droplet?
I
10x?
think probably a mix, because essentially these guys just scoured the literature for any experiment where people had done matched bulk and single cell. So that'll be a collection. I think people used to do that more than now. So it might be that none of them are droplet. My suspicion is that there's got to be at least some droplet in there, but my guess is it's a mix of both, because that was more common in the old days. So definitely some of these are plate-based data sets, but I haven't actually looked through their table of data sets and looked.
Right. So the reason that I'm asking is that, so in the, pseudo-replication paper, as I was mentioning, the first figure is calculating the amount of autocorrelation between in basically different data sets, I think. And I remember that the amount of autocorrelation was higher in plate-based data sets compared to droplet-based, like compared to 10x. And it always seemed to me that like maybe the pseudo-replication bias is more of an issue for plate-based compared to 10x data, but I didn't came across any paper that specifically looked into that.
Yeah, I think I wouldn't go so far as to say that pseudo-replication bias is more of an issue in plate-based, because the correlation between the number of, like, you're always going to have this problem, right? Which is as you increase your number of n, when n is not independent replicates, you're going to have a correlation between n and p-values, which is not real. Like this relationship here should not exist, and once they add random effect size, it goes away. And so, yeah, that's always going to happen, but maybe it's going to happen to a lesser extent with the droplet-based. I agree with you. I've always been sort of the opinion that the nice thing about droplet-based stuff is it's so noisy that other sources of noise get lost in the noise. Like batch effect, I think doesn't matter as much in droplet because droplet's noisy and batch is just more noise, so who cares? It's the reason the meta-analysis methods work so well on droplet stuff. So yeah, that being said, like this more cells, better p-values relationship shouldn't hold. And so yeah, there is like there's always some problem with this. But yeah.
I have one more quick question, but I'm gonna let the others ask their questions first and then come back.
Okay.
Anyone have any questions?
We should have invited Dalaram to come here as the other panelist for a discussion. Yeah.
In general, what I come from this is that you have enough cells or enough samples from different sources, and you do full normalization on the unnormalized count. That should get rid of any kind of bias. Cells from different places, noise not going to affect it. Is that like the correct kind of assumption?
As crazy as that sounds,
Did everyone hear his question?
so his
Brandon?
question was, where did I put that? His question was,
Yeah, I guess it was up to the
if you have, no, it wasn't here. If you have enough cells from enough different samples, you can just log normalize, all your bias has gone away, you can proceed naively, life is good. And that seems wildly naive, but this benchmarking paper suggests that that might actually be true. The things that win are either just doing Wilcox on the data, just no, like a normalized data, but nothing crazy, or doing a meta-analysis combining differential expression testing within each thing on just like the fold change. Those were high performing on droplet data, where it's very sparse, low depth, not necessarily on higher depth or less sparse data. But I think, again, you need like a high number of studies. I think if you've gone and scoured the literature for every glioblastoma paper out there and just combine them all into one data set, you might be able to get away with not doing anything complicated and just doing some tests. But yeah, that still seems wild. I would read this paper aggressively before you necessarily believe that. But yeah.
And I guess also to come on that point where you're talking about double dipping and p-value is inflated and etc. So if you just set a cutoff and you get 0.04 or 0.00001, you just count as significant or you ignore the p-values. That's also a strategy. You focus on log fold change. You don't care if the p-value is very, very significant or just below 0.1. you just consider it as you're not really counting the
So the problem with double dipping is you've lost your type 1 error control, which is your false positive rate. So you are going to get false positives. And you don't know where the line, the threshold to draw for like getting back to that. So if you care about having a false positive rate of 5%, p.05, then you're in trouble because you don't know where to draw the line now at what wildly inflated p-value. If you don't care, because why do we actually care about a false positive rate of 5%? Like there's no good reason for that. We just arbitrarily came up with that. Then it doesn't matter as much, but know that like a lot of those might not be real down the line in your volcano plot. I'm just making waves with my hands, but you're really gonna stop putting transitions on these slides.
I would like to read these papers. Do you think that the p-value is inflated, but there's a correlation between what they're actually significant versus a higher p-value is that better?
So that's what Delaram asked as well, is the rank order of significance conserved? And I don't actually know, but one of the things I always like to look at in these is actually the volcano plot, because like that's effect size versus p-value. And if the volcano plot is like, there's a nice, you know, line there, like the tighter that is, the more I believe it. Once you start getting things where it's like big effect size, low p-value, I'm like, what is going on with this data? Something weird is happening. It's the test. It's something strange is going on, right? And it might be, again, how we're calculating effect size, or one thing's 0, or these tests are sensitive to the expression levels being high or low. But yeah, so maybe in cases where the p-values seem to be very disconnected from the effect size, then the rank order isn't maintained. But in cases where the effect size and the p-values line up nicely, I imagine that the rank order of p-values is true, even if the actual p-values are inflated. Thank you. Delaram, did you have more questions?
Yeah, so basically my question was about how to handle batch effect and pseudo replication at the same time when running differential expression. And the reason is that, so the idea behind these solutions for pseudo replication is that you would use a mixed model. So they usually use a generalized linear mixed model. I think like Nebula and MAST, both of them are doing that. And the idea is that you would incorporate sample information as a random effect to overcome the pseudo replication bias. But the point is that that does not have any effect on batch correction for the sample covariate. And the Nebula paper had a table that indicated that including sample as a fixed effect would actually make the pseudo replication bias worse. So the inflation would be even higher. So I was just, a while back thinking that what's the solution here? Because I don't think the solution is to include sample both as random effect and fixed effect.
No.
If you're trying to handle that. So I don't know, have you came across anyone, addressing this problem? That how would you try to tackle both of these two problems at the same time?
Yeah, this is what I was getting at with the like, isn't it nice that these statisticians have come in and told us that we're wrong, but like none of them pointed out that the way we design the experiments, sample is confounded with batch, or batch is confounded anyway. So yeah, I don't really have a solution for that. I haven't found anything addressing that. All the papers talking about pseudo-replication, either solving it through pseudo-bulk or through a linear model with sample as a random effect term, assume that you know what your samples are and that you don't have batch effects. So yeah, I don't have a solution for that. That's where I was sort of getting at. Either you fix the problem up front with cell hashing, or you just embrace meta-analysis. Were my two propositions, or you try the sandwich estimator thing, which the more I think about it, the more I think you might be right, and it's just randomly getting close to the answer, but not actually being the answer. I don't know.
Yeah, it's hard. I haven't came across anything either, and I've looked for a little bit. But yeah, thank you.
Yeah, we need to do a topic on batch effect correction and integration and all that sort of thing. Maybe Javier can come, because I know they have. Oh, he is. Perfect. Ask Javier. We'll make it his problem. Sorry, Javier, if you're listening. Ah, great. All right. Well, preloaded questions for Javier. All right. I think that's our time.
Right. Okay, thank you, everyone. If you have any questions, you can always email us. And we are putting together the sessions and the readings, and whenever possible, the slides will be uploaded. Thank you so much for coming. We still have 2 more sessions, lectures in this series, which will be up on the website in a day or 2. So thank you and have a good afternoon.
Thank you.